When we usually talk about medical diagnostics, there is this kind of ingrained expectation of, well, mechanical precision.
Right, yeah.
Like, if you break your arm, the x-ray shows a jagged white line, the doctor points to it, and the diagnosis is just binary, it's broken, or it isn't.
Exactly. It's very black and white.
Right. But today, we are diving into something infinitely more subtle for this deep dive. Yeah. And honestly, a lot more profound. I want you, listening right now, to just picture your own immune system.
Which is a fun mental exercise.
It is. Assuming you were perfectly healthy, sitting there listening to this, you probably imagine your immune system is just sort of asleep. Like it's powered down, just waiting for someone to sneeze on you on the subway.
Yeah, which is completely understandable, by the way, because that is exactly how we have traditionally approached immunology. I mean, the vast majority of our medical frameworks, they're built around studying what happens when things go wrong.
Right, the emergencies.
Exactly, an active infection, a raging autoimmune response, or a progressing disease. Mm-hmm. We study the crisis.
So, to use a car analogy, we have spent decades obsessing over how the immune system drives when it's racing down the highway to fight an infection. But today, we're opening the hood to look at how it idles.
I love that analogy, yes.
We're exploring the baseline steady state. And the mission for this deep dive is to unpack a massive, brand new study. It was published in the journal Cell by Babdor and colleagues in 2026. It's called the Immunomicrobiome Study.
And it is a monumental piece of work.
It really is. We are going to explore exactly how the trillions of microbes in your gut are basically the mechanics setting that idle speed, and why this hidden, everyday background chatter dictates whether you'll mount a strong response to a flu vaccine or whether a cutting-edge cancer treatment will actually work for you.
It requires a pretty fundamental shift in perspective, because to understand how that engine idles, well, the researchers couldn't look at sick patients.
Right, that would defeat the purpose.
Exactly. So they recruited 110 completely healthy participants from the San Francisco Bay Area. And they didn't just, draw some blood and do a standard panel. They utilized a comprehensive multi-omic approach.
Okay, I want to pause and make sure we really grasp the weight of that term, because multi-omic gets thrown around a lot in modern biology.
That's definitely a buzzword right now.
Yeah. So if we are mapping a healthy person's baseline, what are the specific layers of data we're actually stacking on top of each other here? Let's go through them.
Sure. So think of it as throwing literally every advanced measurement tool we have at a biological system to capture its entire reality all at once.
Okay.
On the immune side, they use two incredibly high-resolution techniques.
Yeah.
The first is CyTOF, and the second is CIT-SEC.
Let's break those down. What is CyTO?
So CyTOF is mass cytometry. Basically, it measures the specific protein sitting on the surface of individual single immune cells.
Single cells. Wow.
Yeah, down to the single cell. If we think of your immune cells as different specialized workers, CyTOF lets us see exactly what uniforms they are wearing, which tells us their assigned role.
Okay, so CyTOF is showing us the uniforms. Then CI-SEC must be giving us a look at what those workers are actually doing on the clock.
That's a great way to put it. It reads their internal thought process in real time. CIT-SEC measures the active gene expression inside those exact same individual cells.
So we can see what they're reading.
Basically, yes. It shows us which instruction manuals the cell has open and is actively reading at that exact second, so we know who the cell is and what it's preparing to do.
That is wild, but that only covers the immune system side of the equation. To figure out who is setting the idle speed, they had to look at the gut microbiome at the same time.
Right. And they used two more omics for that. First, metagenomics, which sequences the DNA of the entire microbial community in the gut.
So not just a head count of the bacteria.
No, much deeper. This isn't just a census. It reveals the actual genetic pathways they possess. It shows us what kinds of, metabolic factories exist down there.
Okay.
And finally, they used metabolomics on both stool and blood. This measures the actual chemical molecules, the metabolites, that those bacterial factories are actively producing and releasing into your bloodstream.
So we have the cell uniforms, their internal instruction manuals, the blueprints of the gut bacteria, and the actual chemical receipts of what those bacteria are pumping into the blood.
All at the same time.
That is a staggering, almost incomprehensible amount of data for 110 healthy people.
It's terabytes of biological data.
And the most striking takeaway from all that data is that the vast majority of the variation between my immune baseline and your immune baseline It isn't genetic. It's environmental.
The environment is absolutely driving the bus here, and that environment is heavily mediated by the microbiome.
Okay, wait. I do have to push back on the causality here, though. If we are looking at a snapshot of healthy people, how do we know the gut bacteria are the ones driving the immune system? Like, couldn't a naturally highly active immune system be shaping the environment of the gut, dictating which bacteria are allowed to survive and thrive?
That is the classic chicken or egg problem in microbiome research.
Right.
And it's a very fair question.
Right.
The researchers tackled this by looking for what they call concomitant variation, which is where the two systems move in lockstep and then following these individuals over time.
Okay, that makes sense.
But they also leaned on biological mechanics. We know that certain microbial metabolites physically bind to receptors on immune cells and alter their gene expression. So while it's a two-way street, the biochemical evidence heavily points to the microbiome acting as the primary environmental sensor and signaling hub that tunes the immune response.
Okay, let's do a quick recap of this first segment for everyone listening. Number 1, we are studying the healthy baseline immunity, not a sick state. Number 2, the researchers used multi-omics to map single immune cells and gut bugs simultaneously.
Yep.
And number 3, baseline immunity is highly variable, and it's driven by our environment. So I want you, the listener, to think about your own baseline right now. What environmental factors, diet, stress, sleep, do you think are shaping your immune idle speed today?
It's a really interesting thing to ponder because your idle speed is unique to you.
Exactly. So if I'm tracking with you, we're completely moving away from the idea of a dormant immune system. It's less like a parked car that is turned off, and more like a car idling at a red light. The engine is running, pistons are firing, and fuel is burning.
And the RPMs of that idle vary wildly from person to person. But you have to remember, with millions of data points across 110 people, finding the actual signal in that noise is incredibly difficult.
Which means they needed a way to translate all these different omic languages into one coherent story. How do they do that?
They used a mathematical framework called MOFA. M-O-F-A. It stands for multi-omic factor analysis.
Sounds intense.
It's essentially a pattern recognition algorithm. It's designed to find coordinated features across completely different types of biological data.
So it's looking for correlations.
Deeper than just simple correlations. It looks to see if the genetic factories in the gut are dancing to the exact same rhythm as the gene expression in the immune cells in the blood.
Oh, I like that. It's mathematical matchmaking.
Exactly. And this algorithm revealed two major patterns or axes of variation. They call them I4V, which is independent immune variation, and IMCV, which stands for immune and microbiome concomitant variation.
So we are going to focus heavily on IMCV today, right? Because this is the axis where the gut and the immune system are inextricably linked.
That's the one. And the most dominant biological signature in this gut link pattern, basically the loudest beat that both systems were dancing to, was the interferon response.
Wait.
Specifically, tonic and prolonged interferon responses.
Okay, we need to pause right here because this feels like a massive paradigm shift.
It really is.
Because interferons are typically our body's antiviral fire alarms. When a virus invades, cells release interferons to literally scream at the rest of the body to shut down viral replication.
That's the textbook definition, yes.
So the idea that a massive fire alarm is actively blaring in perfectly healthy people with no viral infection,
Right.
that seems totally contradictory.
It would be if it were a blaring alarm. But this tonic interferon operates differently. It is a steady-state constitutive hum of vigilance.
Okay, so it's not a blaring fire alarm. It's more like a neighborhood watch program.
Oh, that's good.
The guards are always walking around, shining flashlights into dark alleys, keeping a low-level vigilance. And this watch program is basically being funded by the gut microbiome.
That is a perfect way to visualize it. And the researchers proved this wasn't just, a theoretical genetic whisper. They looked at the blood plasma.
What did they find?
The individuals who fell into the high IMCV subgroup, so the ones with the strongest gut immune link, they physically had more circulating IFN gamma in their blood.
IFN gamma being a very potent type of interferon.
Yes. And alongside it, they had higher levels of chemokines, specifically CXCL10 and CXCL11.
And chemokines are like homing beacons, right?
You can think of chemokines as literal chemical breadcrumbs that immune cells follow. When a tissue needs immune cells, it releases chemokines, creating a concentration gradient. The immune cells sniff out that trail and follow it to the source.
Okay, so let's do our second quick recap to synthesize this. Number 1, math, specifically the MOFA algorithm, revealed two immune axes, IIV and IMCV. Number 2, IMCV links the gut to the immune system. And number 3, the main driver of this gut immune link is tonic interferon, which is that constant low-level neighborhood watch we just talked about.
Spot on.
So here is a question for you listening. If your immune system is always running this neighborhood watch, what happens when a real threat shows up? Does it help or hurt?
That is the multi-million-dollar clinical question.
Right. But before we get to the clinical side, I want to know about the actual guards on patrol. We know the interferon alarm is humming, but who exactly is holding these flashlights?
The multi-omic data gave them a highly precise roster of the cellular players. In the individuals with high tonic interferon, they saw specific changes in monocytes.
Monocytes. Those are the circulating white blood cells that act as your rapid response team, right?
Exactly. And in this high vigilance state, these monocytes expressed very high levels of a surface protein called sigillase 1.
But here is where the biology throws us a bit of a curveball. Because these sigillase 1 high monocytes are highly vigilant, but the data showed they actually had down-regulated inflammation.
Yes, it's paradoxical.
How does a cell stay on high alert without triggering collateral damage and just flaming up the whole body?
It comes down to a deeply evolved system of molecular checks and balances. Alongside those monocytes, the researchers found highly activated memory T cells.
The veterans of the immune system?
Right, the ones that remember past encounters.
Yeah.
In the high IMCV group, these T cells had high levels of two markers, ICOS and PD-1.
Let's dig into PD-1 because this is a really crucial mechanism. We often hear it described simply as the brakes on the immune system, but it's more sophisticated than that, isn't it? It's more like a rigorous ID check.
That's exactly what it is. PD-1 is an immune checkpoint receptor. When an activated T cell encounters another cell in your body, the PD-1 receptor acts like a molecular handshake.
Like it's asking for a password.
Yeah, it checks the other cell for specific ligands. If the T cell receives the proper handshake, the PD-1 receptor sends an inhibitory signal deep into the T cell, forcing it to stand down and not attack.
Wow.
It prevents this highly activated neighborhood watch from turning into a destructive mob that attacks your own healthy tissue.
So the T cells are fully awake, their flashlights are on, they are looking for trouble, but their weapons are heavily holstered by these PD-1 handshakes.
And that dynamic creates an incredible systemic effect, which we can picture as an inflammation seesaw.
Okay, an inflammation seesaw, I like that.
When the researchers compared the general immune variation axis to this gut-linked IMCV axis, they found that paradox you mentioned. Individuals with the high gut-linked vigilance actually had lower baseline levels of systemic tissue-damaging inflammation regulators.
So things like IL-6 and the TNF-alpha pathways.
Exactly, the classic pro-inflammatory markers.
Because if those run wild, you get chronic inflammation, joint damage, cardiovascular issues, all of that.
Right, but instead of those destructive markers, the high vigilance individuals had elevated levels of a molecule called LAP-TGF-beta1.
And what did that do?
This is a potent regulatory signal. It actively suppresses aggressive inflammatory responses and promotes tissue repair and immune tolerance.
So the baseline isn't just a dial you turn up or down. It's this incredibly complex suspended state.
Suspended animation almost.
We have veteran T cells and rapid response monocytes on high alert, but they are tightly leashed by PD-1 handshakes and bathed in calming signals like TGF-beta1. It's a state of peaceful readiness.
Very poetic, but biologically accurate.
But we still need to answer the biggest question. How are the gut bacteria actually pulling the levers on this seesaw? What is the actual physical link?
They're doing it through chemical manufacturing.
Yeah.
The metabolomic data, remember the stool and blood chemicals revealed 77 distinct microbial pathways and 78 specific metabolites uniquely associated with this balanced high vigilance state.
When you think about the fact that the food you ate for breakfast is currently being fermented into molecules that physically determine how tightly your immune checkpoints are applied, it completely shifts how you view a simple meal.
It makes you respect your gut bugs a lot more.
For real. We can think of these 78 metabolites as a sort of gut care package constantly being mailed into the bloodstream.
And the contents of that package are highly specific. You have short-chain fatty acids, or SCFAs, which act as energy sources for the cells lining your colon, and they have profound anti-inflammatory properties.
Awesome. What else?
You have polyamines, which regulate cell growth and survival, your primary bile acids. But perhaps the most interesting component they found trickling into the blood was LPS, alongside peptidoglycans.
Now that is wild to me.
I know, right?
Because LPS stands for lipopolysaccharide. It is a structural component of the cell walls of certain bacteria. And if you have a massive influx of LPS in your blood, your body goes into septic shock.
It is extremely dangerous in large amounts.
It is a violent trigger for the immune system. So why on earth is it part of a healthy care package?
It's all about the dose and the context. A massive flood of LPS causes sepsis, but a microscopic, steady trickle of LPS crossing from the gut into the bloodstream, it acts as a continuous, gentle nudge.
Okay.
It binds to toll-like receptors on your immune cells and literally induces that tonic interferon response. It's the biochemical equivalent of a drill sergeant quietly tapping the guards on the shoulder every few minutes to make sure they don't fall asleep.
Oh, wow. So it provides just enough stimulation to keep the system awake without ever crossing the threshold into a full-blown alarm.
Exactly.
And this care package isn't just, Amazon Prime shipping from one specific good bacteria.
No, and that highlights the sheer complexity of the microbiome. While specific strains like Hominica brocola were heavily involved, the genetic pathways required to build this entire care package were distributed across a phylogenetically diverse community.
So it takes a village?
It requires an intact, coordinated ecosystem.
All right, time for recap number 3. Let's step back and look at the whole architecture we've built. First, high IMCV means activated memory T cells and monocytes.
Correct.
Second, it also means lower baseline systemic inflammation because of the seesaw effect.
Yep.
And third, gut bacteria send care packages of SCFAs, polyamines, and even a little bit of LPS to orchestrate this.
That's the summary.
So I have to ask you listening, which care package molecule do you find most fascinating, and how do you think your daily diet actually affects its production?
Probably the LPS for me, just because of the paradox.
It's so counterintuitive. But this brings us to the clinical reality check. We have this beautiful map of the steady state. But is this just a temporary reaction to what you ate yesterday, or is this who you are?
That is the pivotal finding that bridges this biology to actual clinical medicine. The researchers gathered longitudinal data. They actually brought these participants back more than a year later.
And what did they find?
The interferon signatures, the circulating cytokines, the specific microbial pathways, they were remarkably stable within individuals over time.
Wow.
This isn't a passing weather system. This is your body's climate.
It is a true, durable set point. Your personal idle speed today is likely your idle speed next year. So how does having a high or low idle speed actually change your medical destiny?
Well, they tested this against 2 very different medical interventions, starting with a seasonal flu vaccine. When they analyzed the data, individuals who naturally possessed this high IMCV tonic interferon state mounted significantly stronger, more effective antibody responses to the flu vaccine.
Well, that makes perfect mechanistic sense. If your neighborhood watch is already awake, walking the streets with flashlights, and the vaccine introduces a harmless piece of the virus, the guards spot it instantly and mount a robust, coordinated response. The system is primed.
Exactly. A high idle speed is perfect for an acute, fast-moving challenge like a vaccine. But then they looked at a completely different scenario. Patients with non-small cell lung cancer who are undergoing immunotherapy.
Okay.
Specifically, a treatment called anti-PD-1 checkpoint blockade.
Okay, we established earlier that PD-1 is the molecular ID check that forces the T cell to stand down and not attack. So checkpoint blockade drugs must be designed to block that handshake?
Yes. They remove the ID check so the T cells can ruthlessly attack the cancer cells.
Right. But how did the baseline affect that?
The clinical data showed something totally counterintuitive. Cancer patients who naturally had a high tonic interferon state at the start of the clinical trial, they did not respond well to the immunotherapy.
Wait.
The treatment basically failed. They only responded if the doctors simultaneously gave them a drug called a JAK inhibitor, specifically atacitinib, which chemically suppresses the interferon signaling pathway.
Hold on, this is the double-edged sword we really have to unpack.
Mm.
Why would a highly vigilant, primed immune system be a massive advantage for a flu vaccine, but a literal roadblock for cancer immunotherapy?
It comes down to the fundamental difference between an acute threat and a chronic threat.
Okay.
A virus is a hit-and-run operation. You want a fast, aggressive response. But a tumor is a chronic, evolving entity living inside your tissue. If a tumor is constantly exposed to a prolonged interferon response, if the flashlights are shining on it, 240-47,
Oh, I see.
it builds blackout curtains.
Precisely. The tumor microenvironment physically changes. The constant interferon signaling drives the T cells into a state called immune exhaustion.
Immune exhaustion. Yes. They are stimulated for so long without a resolution that their internal programming fundamentally alters. They express even more inhibitory receptors, and eventually they just shut down completely. Meanwhile, the tumor upregulates its own defense mechanisms to survive the constant low-level assault.
So by the time the oncologist gives the patient the immunotherapy to take the brakes off the T cells, the engine is already completely burned out from running at a high idle for too long?
Yes, the prolonged interferon has sculpted a highly resistant tumor microenvironment. But by administering the JK inhibitor, they temporarily shut down that interferon signaling.
Ah, so they quiet the neighborhood watch.
Exactly. Which resets the environment just enough for the exhausted T cells to recover, allowing the immunotherapy to actually do its job.
That is a staggering realization. The exact same immune baseline that protects you in one scenario can actively sabotage you in another. And knowing a patient's baseline before treatment could completely change the standard of care.
It moves us away from one-size-fits-all medicine. If we can map a patient's immunomicrobiome axis, we don't just guess how they will react to a drug. We can mathematically predict it and adjust their baseline before we even start the primary treatment.
Let's pull all of these threads together. The old paradigm of a dormant immune system waiting to be triggered is dead. Underneath the hood of every healthy person is a highly active, heavily coordinated ecosystem.
It's a bustling city down there.
Trillions of gut bacteria are constantly manufacturing a complex chemical care package that drifts into your blood, setting an incredibly stable interferon set point. And this delicate seesaw of high vigilance and strict molecular checkpoints dictates how your body will handle everything from a winter flu shot to cutting-edge cancer therapies.
And the really profound takeaway here is that our baseline is not entirely written in our human DNA. It is a continuous, lifelong negotiation with the environment living inside our gut.
Which leaves us with a truly thrilling, provocative question to consider as we wrap up. If your immune set point is highly stable over a year, but we now know it is fundamentally driven by the chemical factories in your gut, could we intentionally rewrite that baseline?
That is the frontier.
Could precision diets, targeted microbial therapies, or engineered probiotics act as a sort of software update for your immune system's idle speed? Could we deliberately lower a patient's interferon set point with a specific fiber diet weeks before they start cancer treatment?
The potential is absolutely massive.
The levers of our immune destiny are literally sitting right there in the microbiome, waiting for us to learn how to pull them. So next time you sit down for a meal, don't just think about calories or vitamins. Think about the neighborhood watch you are fueling and the molecular handshakes you are influencing. Keep questioning the baseline, and we will see you on the next deep dive into the world of complex science.